I want to start by thanking the organizers for this wonderful meeting and the NIH and DZHK. Wolfram and Jay have done a spectacular job at entertaining us with great science and social events, and it's been a lot of fun. I'm not going to talk so much about my own work today because I want to introduce you to a core service that we have that I'd like to have this community benefit from. And so I'll be talking a little bit about RNA therapeutics for heart failure, but a lot about our core service. First disclosures, I'm an inventor on RNA technologies assigned to Stanford and to Houston Methodist Hospital, and the founder of a company that is developing telomerase therapies and a member of some scientific advisory boards and PI on some industry-sponsored research agreements. There is a great diversity of RNA species. Just to remind you, there's many endogenous RNA molecules that make an RNA world that's increasingly becoming apparent that regulates physiology. More recent additions to this species are the circular RNA, for example, and the long non-coding RNAs. Stefan was talking at the morning session earlier about flow-mediated vasodilation. I learned an interesting thing at the RNA Society last week that the flow-activated kinase, the focal adhesion complexes, are assembled by long non-coding RNA that acts as a scaffold for flow-mediated processes. But we're going to be talking today about messenger RNA primarily, and I don't have to tell this group about the biological software that is messenger RNA, but just by way of mentioning, we therapeutic RNA basically is a method for getting messenger RNA into the cytoplasm for translation into a therapeutic protein. And we're all very familiar with this now because of the RNA vaccines against SARS-CoV-2, which were amazingly effective, Pfizer and Moderna's vaccines. But why is it taking so long for messenger RNA to get to the clinic? There's a couple of reasons why. The messenger RNA was discovered in 1961, and it was first administered as a therapeutic in 1992 in a preclinical protein replacement therapeutic. But it didn't get to the clinic until recently, and that's because there were some problems that had to be solved before RNA therapy could become a therapeutic. But those obstacles have been overcome to a certain extent, and this is a new emerging therapeutic field that's going to revolutionize medicine. One of the problems that has been overcome is the, quote, toxicity of messenger RNA, the innate immune activation that occurs when you're administering exogenous RNA. Its short half-life, RNases are ubiquitous. Also, RNA is meant to fall apart. It falls apart in water because of hydrolytic attack on the molecule. Systemic delivery was a problem that had to be overcome. There's some movement toward overcoming that obstacle, and intracellular delivery was an issue as well. And those problems have been, let me go back to that. Those problems have been largely overcome, well, not largely overcome, partially overcome enough so that RNA has become a therapeutic. RNA toxicity has been largely overcome by modified nucleosides, such as pseudouridine and 5-methylcytosine, which were in the RNA vaccines that we all received. And in addition, purification, removing the abortive transcripts and the DNA that's still present is important. DNA RNA hybrids form, they have to be removed because they trigger a pattern recognition response. The short half-life and the systemic delivery has been partially resolved in the intracellular delivery through the use of lipid nanoparticles. And the reason there's so much excitement about RNA therapeutics is that you can target undruggable targets. For example, by comparison to small molecules, enzymatic deficiencies can be targeted. Rapid therapeutic development, we saw that with the RNA vaccines. It's very easy to write the biological software and generate the RNA molecule. It can be personalized. So one can, for example, remove a tumor from a patient, sequence the tumor, and then by comparison to the normal tissues, discover the molecules that are different, surface antigens, and design a vaccine for that patient. Really, that can only be done with messenger RNA. The superior safety profile because it's not integrating into the host genome. As a result of the interest in RNA therapeutics, the big three companies that are developing RNA therapeutics have an amazing valuation for new companies that are just getting into the clinic. One of the reasons the valuations are so high is that these companies have the potential to disrupt the therapeutic recombinant proteins industry. This is a pipeline for Moderna. And if you look more closely at this, you can see in blue are the mainly vaccines that they're developing on the left-hand side. And on the right, there's some other developments. For cardiovascular, there's really only one from Moderna. It's VEGF-A messenger RNA for myocardial ischemia. The company was founded in 2010. They have 37 products in clinical development, many of them in early clinical trials. And they have some significant pharmaceutical partners because Merck and AstraZeneca and Vertex also want to get into this field. So here's the one cardiovascular drug that Moderna has. It's VEGF-mRNA for myocardial ischemia. And they have completed a phase 2A study for safety and tolerability. And in this study, patients undergoing coronary bypass grafting received intramyocardial injections of naked messenger RNA encoding VEGF-A. The patients were then followed for 6 months. So what was discovered with this study is that the therapy was fairly well tolerated, and there were some positive trends. It was a small study, but there were some positive trends for improvements in left ventricular ejection fraction, a reduction in BNP, and walking on a treadmill. So more to come for that. This study was based on work done by Lior Zangi at Mount Sinai, where he showed that naked mRNA injected directly into the heart could enhance angiogenesis. So VEGF-A messenger RNA could enhance angiogenesis and restore left ventricular ejection fraction, and the mice actually had improved survival after experimental myocardial infarction. So what's next for cardiovascular? I think there's a lot that's under development right now. That's in preclinical stage right now, and the idea is that we can help you with your projects, get your idea to the clinic. We've developed basically the roadmap for academic groups and small companies to get to the clinic. We're able to help you design your RNA and synthesize it, purify it, lyophilize it, and we have the analytical tools to validate it, which is GLP grade analytical tools, and we can make the RNA GMP grade for the preclinical studies for FDA approval. And we also have a phase 1 first-in-man facility at our hospital. And we're a small group, a small institute, so we can only do small batches of RNA. So we can do probably 50 milligrams a week, which is enough for a early clinical trial. But we needed a partner to do the phase 2, 3, and commercialization. So we do have a partner now that's licensed our processes and is scaling them up in a 1 million square foot facility outside of Houston, Texas. So this is our institute in the middle of the Texas Medical Center, and the work that we do goes on in the 10th floor and the 12th floor. The 10th floor is where we do our design, and we generate our RNA molecules for preclinical research. And on the 12th floor, we have the clean rooms for GMP synthesis of the RNA. And as I mentioned, we have developed a roadmap toward the clinic, starting with our RNA bioinformatician that helps us design the molecules for codon optimality, for example. And we have scientists that are innovating RNA. I'll introduce you to a couple of them in a moment. And we can help you design your RNA construct, and as I mentioned, we can manufacture and validate the RNA. We can even load it into lipid nanoparticles. We primarily use the precision nanoassembly system, but we have a nanomedicine team. We have faculty in a nanomedicine department that work closely with us to encapsulate the RNA. Since 2015, when we were first funded by CPRIT, we've manufactured for 8 small biotech companies. We support clinical trials with analytical services for when they're going to the FDA. We've consulted for companies and academic groups, and we've designed, well, now it's over 200 unique RNA constructs from over 50 different clients, academic primarily, but also some small companies. And I should say here at this point is that while we're funded by CPRIT now, this whole RNA core, this idea came out of the National Heart, Lung, Blood Institute, Danny Buxton's help with the Progenitor Cell Biology Consortium. With his help, we got some funding to make RNA for the stem cell community. And then when I got to Texas, we expanded it with the help from CPRIT. That gave us enough money to really start a large team and get all this done. In terms of designing of the RNA construct, there are some things that you need to know to make a sequence that's going to work for you. We help people with design with codon optimality, for example. In the 5' UTR, you don't want to have a lot of secondary structure unless it's a circular RNA where you need circular RNA where you need the ribosome entry site, internal ribosome entry site. And 3' UTR, you can manipulate that so that the RNA is expressed in some tissues and not others. But anyway, that's kind of the idea in terms of optimization we can help you with. This is Christopher Brannon, a new recruit. He is doing some brilliant work in discovery of RNA binding sites using this editing approach. He takes makes a fusion protein of the RNA binding protein with ApoBec, which is an RNA editing molecule, and you can then sequence the RNA and find out where the RNA binding protein binding to the RNA. Dan Kiss is making circular RNA. Circular RNA is much more stable than linear RNA because it's immune to the 3' and 5' exonucleases, and it's demonstrably more stable and get better expression with circular RNA. And then we have a nanomedicine team, several faculty that we're working with, and postdocs that are helping us with our encapsulation. We primarily encapsulate using, as I mentioned, the PNI device, which is shown on the left-hand side there. We do make some different flavors of lipid nanoparticles. This is a paper from Circulation Research we published about 2 years ago now. And we incorporate membrane proteins from leukosomes, leukocytes into the lipid nanoparticles to provide for these leukosomes. So the leukosomes are very interesting lipid nanoparticles because they express the membrane proteins from leukocytes. They tend to aggregate at sites of inflammation. And we showed in an ApoE-deficient hypercholesterolemic mouse that we could deliver these lipid nanoparticles to sites of atheroma. One of the things I'm quite interested in, one of our own products that we're developing for the clinic is mRNA telomerase. We've shown that we can restore replicative capacity to human fibroblasts. In this paper, in FASIB 2015, we show that we could essentially double the lifespan of human fibroblasts. You see on the right-hand side the population doubling over time and days after the start of the first treatment. If you give 1, 2, or 3 doses of the RNA telomerase separated by 48 hours, you can dose-dependently increase the population doublings of the fibroblasts. But there's more. It's not just an increase in replicative capacity, we also get an increase, improvement in function. This is some work we just published in European Heart Journal, European Society for Cardiology Journal, and that paper, we used progeria cells cells from patients with progeria to document that we could improve replicative capacity and function of the endothelial cells, the iPS-derived endothelial cells from these patients. The effect was pretty dramatic. You can see in panel C there the improvement in replicative capacity. But we also had substantial improvements in function of the endothelial cells, and also a reduction in DNA damage, which was a bit surprising. In a mouse model of progeria, we were able to increase lifespan. We haven't optimized that therapeutic yet, but it does look like we've got proof of concept. Some other new things in development, some work we did with Bill Poo. Bill actually did the work. We just made the activated YAP mRNA for him, and Bill and his group showed nicely that mRNA could reduce ischemia reperfusion injury and reduce scar inflammation and hypertrophy with improved function in his ischemia reperfusion model. And then we're now working with Eduardo Marban. This is not Eduardo Marban's work, it's work from another group, showing that TBX18 messenger RNA can reprogram cardiac myocytes to pacemaker cells. And that might have some utility for you, for our bioengineering of cardiac muscle. TBX18 can reprogram cardiac myocytes to pacemaker cells. We have to improve on this. Right now, the messenger RNA that we're using is not stable enough. We're going to try circular messenger RNA to enhance the efficiency of the transfection and its effect on the pacemaker cells. This is interesting work that we're doing with Baylor. It's Chagas disease for vaccine against the Trypanosoma cruzi parasite. Peter Hotez has a nice model of Chagas disease in mice, and the Trypanosoma cruzi has a life cycle that includes its incorporation into the heart, causes significant inflammation and heart failure as a result. Major cause of heart failure in South America, and our vaccine, which was the sequence, of course, developed by Peter Hotez, we generated the vaccine for him, clears the parasites from the heart. So we're very excited about that development. So that's our RNA core. I'd love to help you. You can contact me if you have an interest in developing an RNA construct for your bioengineering, and just contact me directly, and we'll help you with that. Thanks so much for your attention.